this is really supposed to be a textbook for undergraduates and graduate students, kind of laying out what the research is on the so-called biology of politics. So a lot of interesting stuff in there, but really like the motivation for me, because I say political participation, is based on a lot of research that we're all familiar with that's studied socioeconomic status, mobilization attitudes, institutions, all kinds of things. But to this point, at least, really haven't focused on biology or genetic influences of political behavior or attitudes as well. And so potentially ignoring genes or biology more broadly could have some consequences in terms of how we evaluate that literature, potentially. So a lot of our theories in politics, political science, is kind of grounded in the role of parental socialization. So especially in terms of partisan attachment or partisan affiliation, but also in terms of political participation. You learn how to be civic minded and all these sorts of things from your family or from your peers. And so the degree to which parents transmit these kinds of instruction is confounded with the fact that they're also transmitting their genes to their children. And so if there is some link between genes and, say, political behavior or just behavior in general, we run a risk of conflating the two and overestimating the roles of social factors. We also don't potentially miss causal pathways through which biology is influencing behaviors. So we completely miss them if we don't study biology, and then we also don't fully characterize theories that we already have. If, like, for example, there are, we're mistakenly thinking that socialization is actually the reason for what we're observing. And then finally, we might be missing a possible source of heterogeneity in terms of environmental stimuli or policy intervention. So this is kind of one of the arguments that people study personality also make. So not everyone is going to respond to some kind of stimuli the same way. So some people may have a very strong response, some people may have a weaker response. And so without kind of having some source of genetic influence in our analysis, we might be washing out these heterogeneous responses or treatment effects. And so from a practical standpoint, a lot of things like voter turnout, the variables that we've been studying to this point actually account for surprisingly little variation, a little amount of the overall variation. So, a huge model of voter turnout, 32 factors, only accounts for about a third of the variation of turnout. So it could be that we're missing a fundamental or an important source of variation, and potentially genes or genetic factors might be that source. And so, we know that they're from behavior genetics literature that genes have been shown to be relevant for all kinds of traits that we know are related to political behavior or political attitudes, like personality and also social attitudes. So it might be that might lead us in the direction of actually having a more complete picture. And so really the fact that genes matter for all kinds of behaviors, we shouldn't think that politics is necessarily different from those. And so we should probably it would make sense that they would matter for political behavior as well. And so, as was alluded to earlier, twin study design was kind of the first foray in this, or the first kind of tool that political scientists employed to study the influence of genetic factors on political traits. And so the idea is, for those who are not familiar with the kind of setup of the twin study, if you are familiar, I apologize, but identical twins or monozygotic twins share 100% of their genes, and dizygotic or fraternal twins share on average 50% of their genes. So the idea is, if we can compare the similarity on traits that we care about of MZ twins and DZ twins, we can kind of back out the role of genes. If we make a very critical assumption and a kind of a controversial assumption, this equal environments assumption that the environments MZ twins and DZ twins face, it's a little bit more nuanced than that, but the environments they face are the same. So if we make this assumption, I'll show you the math in a second. then we can identify the model. So what this twin model does, it's a latent variable model where we assume there are 3 latent factors: additive genetic factors, shared environmental factors, and unique environmental factors. And so we have this additive model, and another assumption where we have the weights on the 3 latent factors. And so in our case, the Y would be a political trait that we care about. And so the way to interpret A, C, and E is A is just this the influence of adding up all of the influence of genes additively. And then shared environmental factors are things like early socialization, early schooling, political association. So the things that both twins have in common. So that's the shared environment. And then unique environment is just the residual, and in this case, it includes measurement error as well. So then this is a bunch of math. If we make assumptions, essentially we're left with a system of 3 equations, 3 unknowns. So that means we can solve for the 3, and we get these nice A squared, C squared, and E squared. So A squared is what we know as heritability. So it's just the share of the variation in the trait that can be attributed to additive genetic factors. And then we have 2 other, based on our model, we have 2 other buckets that the variation can go into: the shared environment and then unique environment. So ACE. And so this is the model that a lot of the early studies employed, and it's been used to study a variety of traits. So there's been special issues in the Journal of Theoretical Politics. There's been a special issue in political psychology. Behavior genetics had a special issue on politics. And so a ton of studies have looked at things like political attitudes. That's probably one of the more widely studied traits. Then behaviors like turnout and overall participation, which I showed you before. And then also interest in politics, whether or not you view voting as a civic duty, political efficacy, political knowledge. So there's a whole host of them. And estimates of the heritability, the proportion of the variation in these traits that you attribute to additive genetic factors tend to range between 0.3 and 0.6. So about 30%, between 30 and 60%. And so here's kind of a little, here's a figure where blue is the heritability, the A. Green is the common environment or shared environment, and then red is the unique environment. So this sums to 1. So the blue is the proportion of the overall variation that is you can attribute to additive genetic factors. So that's the heritability. And so you see for all of these measures of political attitudes in countries like the United States, Sweden, Denmark, Australia, using all kinds of different measures of authoritarianism, indices of Wilson-Patterson index, the top 2, the left-right, the traditional left-right scale. So you see there's as I said in the last slide, between 30 and 60%, but there's also relatively little shared environment if you based on these results. So this is also kind of another surprising thing that's come out of these studies is this doesn't necessarily it's not compatible with the idea that parental socialization, because that would fall into this shared environment category. Parental socialization is a big deal. So its unique environment is obviously the biggest, but then heritability is bigger than shared environment. If anyone has any questions too, feel free to chime in. I don't know what the protocol is, but I definitely ask. So here are measures of political predispositions and measures of participation. Again, Denmark, Sweden, United States. And so knowledge, how often you discuss politics, turnout, overall participation, efficacy, things like that. And so the striking thing, and this was originally reported in Alfred Funk and Hibbing, they found the same thing, is that in terms of party identification, whether or not you identify as a Democrat or Republican, not the strength of your identification. So am I a strong Democrat or Republican? Am I kind of a leaner? Those don't have as much common environment, or not as much as attributable to common environment, but party ID is, right? And so Alfred Funk and Hibbing make the case, and others have too, that it's that is where you get your identification is where you get or your parents are is where you get your party identification, which is consistent with the theory. And so this does in this case corroborate kind of early theorizing that parental socialization is a big deal. So twin models, there are a bunch of extensions that have also been used by political scientists. You can look at additional sibling types. So you have 3 equations and 3 unknowns because you have MZ and DZ twins. So you can relax those assumptions by including additional sibling types like half brothers, cousins. You can also include parents if you want to ask additional or other types of questions, like whether or not there's if people are mating for specific reasons. And so that's one thing that's been done. Also, longitudinal models. You can look at the heritability of traits over time. And so there's a recent paper by Atimi that shows that the heritability of political attitudes is 0 when you're in an adolescent. But then once you're older, their argument is once you leave the house and you're kind of free to do what you want, then the heritability becomes much larger. So, and then it actually middle kind of when you're in your 30s, then it approaches the levels that we kind of see from the alpha function and other papers that came after that. So people have looked at longitudinal models with twins, bivariate models. This is something that I have worked on, where I'm interested in seeing if political participation and personality traits or cognitive ability have some shared source of genetic variation. So it could be that the personality and cognitive ability are mediating the relationship between genes and participation, or it could be that they're just both influenced separately by the same set of genes. And so it just so happens that the 2 are correlated because of this genetic, the same underlying genetic architecture. And then gene-environment interaction, the assumption that I showed, the linear assumption, it doesn't necessarily have to be the model that you use. You can see whether or not environment interacts with genes. And so another Atimi paper looks at the interaction of life events with genes, and to see whether or not they influence their difference. Essentially, the effect of genes moderate political attitudes. And so that's an extension that folks have explored. And so really, twin studies are great because they give us a starting point. They answer the question, is there something there to look at? So they tell us whether there's something there, and they kind of give us an idea of how much, in terms of like how much of the variation can we attribute to genetic factors. So that it's definitely very important from that standpoint. But really, the next question is, and I think where the literature is going now, or started to go, is how genes matter, or why genes matter. So what are the specific mechanisms by which genes influence behaviors or attitudes that we care about as political scientists? Because they're not going to influence them directly. So one way to do that is the bivariate model that I talked about a minute ago, trying to put more complexity into the relationship that you're studying. But then what I know a lot of people have done is try to use molecular genetic data. So actually going and measuring genotype and seeing whether or not I can find a specific gene or a set of genes, and then that will give me, ideally, shed some light on what the biological mechanism is that's at work. At least that's the hope. And so there are also other advantages if we can if we think that the genes potentially confound some relationship between say personality and political participation, or just there's something that we need to control for in our models, then we can use genetic information to actually, in non-genetic studies, to kind of control for the fact that it could be that genes are playing some role in the background. And then again, as I mentioned, heterogeneous treatment effects. That's something I can get a better handle on if I have specific information. And so this has become more feasible because the cost of genotyping has dramatically declined, and it's getting cheaper and cheaper, even though it's still kind of expensive. And so a lot of studies that have been done for medical purposes are working with political scientists, after they've already collected the data, to try to enrich these data sets to use for our purposes. So a lot of social scientists, I should say, have access to this data that they never had before. But then major studies like the Health Retirement Study and the National Longitudinal Study of Adolescent Health are releasing, along with their survey, very extensive survey data, genome-wide data. So they're making it available to social science researchers, but it is still limited. Even though it's becoming more available, it's still limited. And one of the major challenges is that really identifying a gene is kind of not even half, or identifying a variant is not even half the battle. It's understanding what in the hell that your result means is incredibly challenging. So like you get excited about this kind of stuff, but then you realize, oh my God, I mean, I have a pretty big task at hand, even if I find a result, because then I have to try to figure out what it is that I found. And the fact that any political behavior is going to be influenced by many, many, many genes, which are likely going to have very small effects in and of themselves. It's going to be hard to detect them using the techniques that we're kind of have available to us now. And so the first, I think, foray into this, again, that's the 2 times for foray. For those of you guys playing the home game, that's 10 points. The first studies using this molecular data took a candidate gene approach. And so the idea is that you've selected a gene because you think that there's some theoretical reason for it to influence the trait that, or be related to the trait that you're studying, or it may just be that other people have found results that you think are in line with what you're studying, and so you kind of borrow the empirical result to say, or not borrow, but base your inquiry on the empirical result of somebody else's report. And so a lot of studies, or the political science studies, are focused on dopamine and serotonin genes, primarily because they're related to things that we think might also be related to political traits. And so one caveat is that many of these candidate gene studies have medical genetics is kind of plagued with these failed replication studies. So what happens is somebody reports a result, and then somebody else goes out and tries to replicate that result on a new sample, and it doesn't work. And so then there have been so many of these that it's really changed the kind of the way people view candidate gene association studies. And so another approach is the genome-wide association study, which requires no knowledge of a pathway. So you've genotyped hundreds of thousands, if not millions, of variants across the genome, and you just want to do a data mining exercise to see if any of them are associated with. And when I say association, essentially you're just regressing whatever dependent or whatever political trait you're interested in on a genetic variant, and then control for a couple of things, and you want to see whether or not there's a significant beta coefficient. So you've got 500,000 to 1 million, and then you want to see if there's anything that pops, and then you'll go back after the fact and try to understand why it is that there's this, why you found this relationship. So those are 2 different approaches using this data. There are some others too, but those are the 2 main approaches that, at least in politics, researchers have embraced. And so there have been a couple and published in our journals, Journal of Politics. So one was with James and I, which was the Evan Charney had a piece in the APSR talking about that paper. Then we had another paper in the JOP that was looked at partisanship and a dopamine receptor gene. And then there was another one, another dopamine receptor gene, and ideology. And so all 3 of these studies use data from the National Longitudinal Study of Adolescent Health. And what we have, we had a response to Evan's piece that's going to go into the APSR, where we conducted, we attempted to conduct a replication of both of our original findings And we found that one of them we could replicate and the other one we couldn't replicate. So we had a much bigger data set this time because AdHealth released a new batch of data. And so we had a much larger sample to try to see whether or not our original results held up. And so, as I said, one did and one didn't. And so that's not uncommon. There was some of my colleagues published a paper in Psychological Science where they looked at public associations of IQ. So there were 13 genetic variants in 10 different genes that have been published, and they tried to replicate them using 3 very large samples, and they couldn't replicate any of them. So this is just another example of what has already been kind of hashed out in the genetics literature, but social scientists, our example and this example is important. That this is candidate gene studies without proper replication are problematic. And so I'll kind of get back to why we think that is. But so we also tried to, in a recent paper published in the Proceedings of the National Academy of Sciences, and then some other stuff I've added in here, we tried to do a genome-wide association using some Swedish twin data on several political and economic traits to see if we could find it. So we have the survey that we're using actually is a very detailed survey of political and economic questions that we sent out to the Swedish Twin Registry, and so we have about 10,000 responses, and a subset, about 4,000 of those were genotype that we used in this analysis, and so they're older, and yeah, so about 4,000 people. And so what we did was we asked them a battery of questions to elicit their policy positions, and after factor analysis, we came up with 5 dimensions of ideology: immigration, economic policy, environmentalism, feminism, and international affairs. And we also did another factor model to come up with an index of 8 different acts of political participation, kind of standard questions: whether or not you contacted a politician, boycotted a good, made a financial contribution. So that's another one that we studied. And then in a separate research, a sample that I used, folks that I work with at the University of Minnesota, they also have a twin sample that they have genotyped. They have genome-wide data on for about 8,000 subjects. And so just to do another genome-wide association study, I looked at validated voter turnout. So validated meaning matched the people in their study to Minnesota state voter records, which are publicly available, to see whether or not they voted in the 2010 election. And so ended up having about 5,000, just under 6,000 matches. And so the matching was consistent with what the overall turnout rate was. So it's pretty confident that it's good matching. So then what I did was, so the political behavior from the Swedish study, political participation, 5 measures of ideology, or 5 dimensions of ideology, and then whether or not they voted in, whether or not Minnesotans voted in the 2010 election, did a genome-wide association for each of those traits. And so both the Swedish study and the Minnesota study had very similar, there's about 600,000 genetic variants that were typed for each of the subjects in their sample. So it's a huge number of regressions. And so after correcting for multiple testing, you have this very high threshold for what's significant. And so none of the measures that I looked at were significant once you account for this multiple testing. So in the 3 measures in 2 different samples, no genome-wide association, or no significant genome-wide association. And so the paper that some of this is in kind of goes into more detail, but the real point is, or the real, I think, culprit in not only not finding anything in these genome-wide studies, but also finding false positives in candidate gene studies, is that there's just not enough power to be able to find very small effects in the case of genome-wide significant or genome-wide association studies, but in the case of candidate gene studies, you're just getting, you're getting something by chance. And so here's a power graph that shows the colors are how much of the overall variation each SNP or genetic variant would be responsible for, or not responsible for, how much of the variation could be accounted for by a SNP. And so the black is 1%, so that's huge. And like height would, or something like height would be less than 1%. So 1% is really, really big. So it's probably, what we could expect is somewhere like the red line or maybe the yellow line, with something much smaller. So this shows you, if you want, if you power of 80%, 5% or an alpha of 0.05 after alpha of 0.05, you're going to need massive samples to have a relatively big, if you're the variant that you're studying has a big effect size, but like more realistic ones, you need very, very large samples, and none of the studies that have been published to this point have the power that's required to be able to say something meaningful. And if you look at genome-wide association studies where you have to make this multiple, you have to correct for multiple hypotheses, it's much worse, right? So again, well, it doesn't look at that, but if you look at the scale of the x-axis, you need tens of thousands to be able to find, to be able to detect these very, very small effect sizes. So that's kind of like one of the biggest problems, challenges that this next kind of phase, if we think that doing these types of association studies is the next phase, then that's kind of the big challenge. And so colleagues in economics have started to try to create, well, they've created what's called the Social Science Genetic Association Consortium. And so what we've done is piggyback onto these very, very large health consortiums and ask them if we could essentially use their genetic data, and then if they're willing to participate, help us collect new information that political scientists or social scientists would be interested in, or at the very least, let us study things that they're not necessarily interested in that they've collected that we are. And so one of the first analyses that actually is getting ready to be published is a very, very large study of educational attainment. And so using over 100,000 subjects, they were able to find some interesting results, but it took very, very large samples. So I guess the point is that if we're serious about doing this type of work and the power that's required to do it, we don't have, this is something that we're going to have to embrace or try to become a part of. And so then there are alternate things to do that we are adequately powered with the types of sample sizes that we have with comprehensive genetic data. And so these are things we might want to explore, and the paper that I referred to earlier kind of tries to do a couple of these as a proof of concept to see whether or not we find any interesting results. And so one thing we can do is, using the genotype data, is get a direct measure of heritability. So heritability, what I showed you before with the twin studies, requires that you make these assumptions. You're looking at MZ and DZ twins because you know how they have known relationships, and you can leverage that to back out genetic information. An alternative approach would be just to use a direct measure of how related people are genetically, and whether or not that is, or how strongly that relates to how similar we are on some trait that we care about. So kind of cutting out the need for twins, but kind of using a similar approach. And the nice thing about this direct estimate of heritability is it doesn't require the equal environments assumption. So if you get a similar result from this, then that tells you maybe something about twin studies, but you don't even have to worry about the equal environment assumption at all. And then another thing we could try to do is see whether or not the genetic information that we've collected is predictive of some trait. And so health researchers have used these risk scores to see whether or not you can predict predict a disease or some behaviors as well, but mostly diseases. And so we can see whether or not this would also work for political behavior, or for behaviors in general, political behaviors more specifically. And so for the estimate of heritability, we just want to see whether or not all the SNPs jointly, how much of the variation we can explain in a particular trait. And so we can directly estimate that using some interesting techniques. And so this Gremel approach it represents a lower bound of heritability because you're only capturing the information from the genes that from the genes that you genotyped. So you may not have all of the genes, so you may not be capturing all of the heritability. But this has been used for cognitive ability. It's also been used for personality traits. It's been used for height. And the results tend to find slightly lower estimates based on this approach than what you find in the twin study. And so again, it could be that you just don't have all the genetic information at your disposal, or it could be that the twin studies, the estimates from twin studies are inflated because of maybe a violation of the equal environments assumption and things like that. It's unclear yet. So what we did was see whether or not we could do this for the 5 dimensions of ideology using our Swedish data. And so we have this from the twin studies, we have estimates of heritability, and then this is the estimates that we get from this Gremel approach. And so you can see for some, the measures are relatively noisy because we don't have a very large sample, but it's dramatically different. But then for others, it's kind of similar to what other studies have reported. So the question is this just because of noise? If we had a bigger sample, we'd have a more precise estimate, or is it the twin studies are giving us incorrect results? But again, so this technique can be used to give us a better estimate of the overall the contribution of genetic factors to variation in political traits, without having to worry about getting into an argument about the equal environments assumption. So we also tried to construct a risk index. So essentially taking from these genome-wide association studies all the regression coefficients and just adding them up and making them into an index and seeing whether or not we can predict out of sample using that index. And so that's what we did for the measures of ideology, and we found that we couldn't predict very much. So none of them were significant, and even the biggest one was a very small percentage of a very small amount of the overall variation out of sample in say attitudes towards feminism. So this doesn't necessarily doesn't look like this technique unless again we have more data that as of right now it holds much promise. So kind of the conclusion of this, the promises and pitfalls. This is I guess a little bit of both. Is that this shows that, or at least the analysis that I showed you the sum of today shows that political traits are likely influenced by many genes that have very small effects, and we're going to need very, very large sample sizes to be able to detect them. Even if we have a candidate gene study and we're looking at one particular SNP, we're going to need pretty big samples because we shouldn't expect some kind of like gene for voting or gene for that. It's going to be a bunch that have a very small effect. And then we also have a limited ability to predict traits, which is unfortunate because it would be nice to be able to use these types of indices to control for the effect of genetic factors in non-genetic studies. And also, economists are particularly interested in using genes as instruments to try to get more leverage on some causal relationship. And so, I think it's a little too early to be able to say that either one of these is based on the data that we have it's going to be a while before we're able to do this. So our response and that's going to go into the APSR is that this is really now is the time to kind of take a deep breath and think about what are the challenges, or I guess just understand the challenges, and then take that into our decision-making process going forward about what we want to do and what we need to do to be able to do what we want to do in terms of this type of research. And so just, I guess quickly, some other areas. So genes are the area that I study the most. Also, there's neuropolitics, hormone. People have studied hormones. People also studied physiology. And so some examples of neuropolitics. There's researchers at my school, Amodio and John Jost, wanted to see whether or not there were differences in the way liberals and conservatives evaluated conflicting information. And so they had essentially a measure of cognitive flexibility. So they had folks do this go no-go task, and found that in fact liberalism was had a higher response in the anterior cingulate. So this was kind of like the first foray into neuropolitics, which kind of were followed by several other studies. That there was a recent study, interesting study, that asked whether or not there were brain structural differences between liberals and conservatives. And so what they did was they analyzed brain volume, and so they found that liberals had a larger ACC, which is consistent with Amodio findings, and then conservatives had a larger amygdala. And so then there's a bunch of other descriptive type studies where they have some measure that they're interested in. There's one, Darren Shriver and a bunch of fellow researchers from UCSD wanted to see if Democrats and Republicans had different activations when processing risk. And so lo and behold, we found that. But essentially, like all of these are looking for a correlation between a brain region, either activating or brain volume, and some political shape. So it's not necessarily to the point where theories are being tested. Hormones are also something that people have started to research more, and in particular looking at oxytocin, vasopressin, and also testosterone and cortisol. And so seeing whether or not these different oxytocin or testosterone levels actually are associated with different types of behaviors or different levels of behaviors. So oxytocin has been linked to trust and affiliative behavior, testosterone aggression and risk-taking, and cortisol to stress. So the degree to which affiliative behavior, if you're a Democrat or Republican, or risk-taking or aggression matters for politics, that's essentially what early studies have tried to figure out. And so Rose McDermott and her co-authors looked at testosterone in war games and found there was a relationship between engaging in unprovoked attacks and testosterone levels. French and co-authors looked at cortisol levels and found that there were higher levels of cortisol meant people were less engaged in politics. And then also the physiology. I'm sure a lot of folks are familiar with the Oxley paper that was published in Science not so long ago. And the idea is whether or not there are physiological differences that may be meaningful for political behavior or attitudes. So an early study looked at Mutz and Reeves looked at how people responded to incivility in political discourse, and also Oxley looked at whether or not physiological responses to threatening stimuli were different for Republicans and conservatives. And so they were shown these threatening pictures, and then they were given a skin conductance test or a startle eye blink test, or actually no, just skin conductance. And so there was a big difference among liberals and conservatives in terms of their responsiveness. And then they also did an eye blink test to see how they reacted to a blast of unexpected white noise. And again, people, liberals or conservatives, had a much stronger reaction to this aversive stimuli. And so the implications for politics, a lot of these are in their infancy. There haven't, in all these domains, there haven't been a lot of studies published yet. But they're really, and a lot of them are really just correlational studies. It's hard to know whether or not there's something causal going on there. But a challenge is that I think the book talks about a little bit is trying to link these intermediate steps with political participation or measures of politics, and then also say back to genes. So trying to come up with a more complete causal story using all these different techniques is going to be another challenge that this literature is going to face, trying to put the pieces together. And so one example, a toy example, could be how it would fit. We know oxytocin is associated with higher trust. So people have been given exogenous doses of oxytocin in the trust game, and they tend to be more trusting. We also know that receptors or genes that code for oxytocin receptors are associated with empathy and trust, just based on association studies. We don't know that, but there's evidence suggesting that. And then also these variants have been related to amygdala volume or volume and activation. So it could be that there is a story here linking it all together. Now this is a toy example. I have no idea if this is the case, but this would be the type of integrated approach that would ideally, kind of put a story together and be a more satisfying explanation of how biology might actually influence traits, proto traits. And that's it. Any questions, obviously, or comments?
Can I just ask
Math?
a very specific question?
Sure.
Yeah, so we gotta open it up.
Okay.
Do you want to field your own questions, or do you want me to? Or?
Do you mean someone else will answer them for me, or?
That's cool. That. So you want to field your own?
Sure.
Liberal and conservative, is that based on party affiliation, or is it do you have a test of liberal and conservative idea?
So they're both. So some measures are based on partisanship, but most of them are based on asking a battery. Alfred Funk, the paper Alfred Funk and Hibbing did, there's a Wilson-Patterson battery where you ask, do you support a number of policies? It could be 30 policies. And so based on your support for those policies, you put on this left-right dimension. It could be more than one dimension based on a battery of policy questions, with the Swedish example. It could also be just I ask you, where do you place yourself on a left-right scale? Left being liberal, right being conservative. So there's a bunch of different approaches that have been used to try to measure ideology.
There's so much in that. For example, you have questions that relate, say, to pacifism versus non-pacifism, or solving problems through. Is that part of liberals are more pacifist, while conservatives are more?
So one of the items in the Wilson-Patterson battery asks you specifically about pacifism. So it's not a nuanced question like, how do you feel about pacifism? It's more yes, no, and then you say the assumption would be liberals would say yes to pacifism, conservatives would say no. I mean, I don't. That would be the answer. That would be what some people would suggest.
Maybe come back to it.
Okay. Well, so a couple parts. So even the genetic information that you're using, that has measurement error too, because you're taking a swab and then you're sending it off to a lab, and the lab has to. It's not precise in terms of the. It's not known with certainty what, say, the genotype is. It could be they have to make a call. There's 3 different possibilities. It really looks like this one. sometimes it's very clear that it's this one, other times it's like, ah. And so even that is, there's a problem, but I agree, the dependent variable is much more noisy, and that's part of the reason too, contributing to this, the power issue is not just about, the fact that genes have a small effect. The more noisy your measure is, then the more power you're going to need to get, cut through all that noise. So that's definitely contributing to it. And then when you have like complicated measure or complicated things that you're trying to study, like ideology, then it even makes it more complicated. But in terms of accounting for measurement error, some twin studies you can, like, so if you have test retest information on traits that you're interested in, you could account for measurement error in a model using the test retest information. Sometimes twin models will have multiple instances of asking the same question. So I could ask you now, a month from now, 2 months from now, put that all in the same, kind of a factor model, and then that would also, again, account for the possibility of measurement error. But unfortunately, not a lot of people collect that type of information, especially in politics. There's not a real strong kind of norm of collecting test retest data, or we tend to think that I ask you a question now and I ask you again in 2 years, and if there's movement, it's because of this kind of chain evolution or whatever. So I agree. I mean, it is very difficult to when we have the measures that we have to try to get a precise kind of estimate. Yeah. Well, so I mean, part of it is they're trying to get back to the actual. See if I can find it. Yeah, that's it. Yeah. So this is what you're talking about. So the difference between these two.
Yes. Yeah. I was thinking more. Yes. Okay. That's. I was driven more by your previous slide. How big sample for a gene that had where variance in account 1,000.
Yeah, so if we're looking for this technique just tries to say how much of all the variation in this trait could I jointly account for using all of my SNP data. So it's essentially like this big random effects model where I have every one of the genes in my equation. It's not that's not exactly how it does it, but it would be that kind of idea. Right, I account for everything. And then that R squared would be, how much genes are capturing. Whereas the GWAS, like when I'm looking for just a single, like that coefficient on a regression, the regression coefficient, telling me how big of an effect that SNP is having on my attitudes towards immigration. Those are kind of a little different. This approach doesn't require the same type of power.
Eventually, you need some model in which you have 4,000 variables, collect enough genetic information anywhere close to that.
Well, yeah. Well, no. So in terms of like finding, so one of the like these genome-wide approaches, like where you're interrogating 500,000 variants one at a time, and so in principle, if you had all the genes, you should be able to add up the effects, right? Those beta coefficients, and it should sum to what you get from a twin study. And so there's this this big finding is the missing heritability. When you go and do these GWAS studies, you don't find it just didn't work, right? And so the question is, well, why didn't that work? And the problem is you're estimating these beta coefficients that you're getting are so noisy and so imprecise that that's one explanation potentially. This gets away from that because it just says I'm going to. It's essentially like a more complicated technique, but it essentially is saying I'm going to essentially regress all of these in the same equation and see how much I can account for. So this would be the same as a twin study, but if I had all the genes that are in the twin study, although I mean it could be I'm not capturing interactions, gene interactions, all kinds of things like that. Yeah, that.
You do get this does account for the sort of some versions of conditional effects.
Yeah. Yeah, and so there could be other things, other factors at work, but you should get pretty darn close if you had the genotyped a bunch, millions and millions. I don't know how, but if you had them all in there, this should give you these two. If you believe the twin study result, these should be the same. And then in principle, if they were different, then you would say, it could be the violation of the equal environment assumption, could be all this other stuff. So that's why I think this is cool, is because if there is a debate within the literature that twin studies are junk because they're within our literature, twin studies are junk because they make all these crazy assumptions, and so what you get out in terms of this heritability estimate is completely wrong. It's way bigger than in reality it is. And so this at least could, if we're not powered to do the GWAS studies, this could kind of help us resolve the debate of what really is, how much variation can we put in this genetic bucket? It's not what I thought you were going to say in terms of under the hood. It doesn't tell. It gets me back to the original problem. I don't know if I don't know which genes are doing the work, then that doesn't tell me anything about the underlying biology that might be responsible. I mean, assuming that if you knew the gene, you could then kind of figure out the story, which is still a leap because it's really hard. But at least the promise is that will give me some information and kind of point me in a direction, maybe a novel direction. I never thought about this. This is something political scientists have never studied before, and here's a new theory and that kind of stuff. Then you're famous.
Can you go back to slide 15? It's the slide where you have the different amounts of heritability, shared environment, and then the third category for the different traits.
So is it the
Where you have authoritarianism. Yeah, the slide right before that. There.
yeah.
So what strikes me in the slide, I'm curious what your explanation of it is. You talked about in the twin studies at least you have between 30 and 60% heritability. And what I noticed here is on some of the same, especially left right as well as authoritarianism, the heritability on the exact same thing is quite different from the countries. So I'm wondering what you think accounts for that. Is it because in Sweden genes matter less to ideology, which is strange? What's the explanation for that? Why does it get different from countries? Is it all noise?
So one of the things that this heritability estimate, like an R squared, it's really specific to the data that you're looking at. You can't ideally, or not ideally, you shouldn't kind of extrapolate to other cases. Although you find across these different contexts, you find remarkably similar heritability estimates. Left right in terms of Sweden, I think is just maybe left right is not a very good measure of, given the fact that we were able to decompose ideology into 5 dimensions. It's not doesn't really have as much resonance. So maybe it is just capturing noise, or maybe within the context. I mean, ideally, that would be the thing that you could do is, you have this gene environment where the environment is, say, the institution, like the political institutions. And so being able to do that would be really cool. But, it's kind of, I hesitate to do it because I don't know.
The left-right measurements, which I think are different in authoritarianism measure, quite different.
These two?
Yeah.
No, I think these are, I'm pretty sure these are similar. These are very similar. So this, I mean, it could be the context. Yeah,
The
that's.
heritability of authoritarianism in the US is twice as big as the heritability of
Yes. Yes. Yeah. I mean, so there are, I'm sure, there are confidence intervals. So it's, but yes, I mean, it's, you take the mean or the point estimate, yeah. So again, that would be something interesting to do. There are also studies in the U.S. that have looked at heritability over time and specifically changes in context. So like the heritability of smoking, when very early on, when soldiers were sent to war with cigarettes in their packs, then the Surgeon General, then this, then, and so you see very clear changes. And then also women and men, you see like it's there's a stigma against smoking among women at certain periods, and then that stigma's gone. So that is very very interesting, but it's kind of I think in this case maybe a little bit limited, but yeah.
Although I interpret the difference between US and Sweden differently, I think that left-right scale is really extremely strong. It's very easy to measure people close to dimension two, and I'm not sure because but I wanted to know also how you explain the differences and to give an answer. And I also have another question because I don't know if you obviously don't know it, but the twins are getting tired of being answered questions, right? So is there like a almost like a prisoner's dilemma or researcher's dilemma here that we cannot ask them? I mean, we cannot ask them all the questions we want to ask them, and we already see signs that they don't answer correctly. They're not taking it as seriously as they did before. Have we seen that?
So I've actually spent like more time than I'm willing to admit around like not twins, but studies of twins where they come in and participated, and I've gone to a couple of twin festivals where they come and participate in our research. They come to get money to go to like the food area there, but they are incredibly like committed to actually not they're not monolithic, but I encountered a very strong feelings that they realize that they have they're in this unique circumstance and they want to contribute to the science. I agree though, they do get they get asked a lot, and there is the studies that I've been that I've been a part of are very careful not to push too hard. So they are resistant to send surveys, very very often to the twins. They don't really want to tax them with long batteries. They make sure that they're compensated pretty well. So I'm sure, like anyone else, they feel fatigued too. But I have been struck by their kind of like norm of wanting to contribute. MZ twins more than DZ twins, so that maybe violate the equal environment assumption, but yeah. Yeah.
I wanted to know, actually, I have two questions. So the first one is, has anyone ever looked at so twins, the assumption is that identical twins share 100% of the genetic information. Now, is that really true, or is there some variation? I assume there is some variation, not all twins like relate to each other 100%. And if there is some variation, has anyone ever used that variation within twin pairs, or between, sorry, rather between twin pairs to make further conclusions? So that's my question.
So the MZ twins are unique, and there are alterations to the genome after conception. So as you get older, there are all kinds of things that can happen. So they're used increasingly for that, to study the differences that must have related to environmental exposure and things like that. So and there are more sophisticated types of analyses that they're being like in a molecular genetic context that they're being used for. But not in terms of none that I know of in terms of politics. But there are, yeah.
In the medical area. Mm-hmm. So that would be maybe another thing to explore.
Time's up. Can't go on. No more questions.
I had actually one more question. Okay.
Oh no. No.
I actually had a similar question as Stuart. In a way, I saw your presentation as
twin studies, but when you follow up on other methods, you just don't find what you want to find. But I kind of want to ask you, like, what keeps you going in that direction? Is it the result that you showed us in foreign policy and economic policy, there where you find, you know, the most kind of hope, I guess, or I don't know how to phrase it, that genetic sources really do matter and that it can be replicated with this new method? Is that what keeps you going, or what kind of keeps you going, I guess, in that direction?
Lots of coffee and so other results I didn't show. So for again, like my main kind of the trait that I care most about is participation or like turnout, whether or not I participate in turnout. And so other analyses that I've done for all Minnesota and Sweden looking at participation, actually there's not as big a gap as there is for the ones that I showed in terms of the twin result and the actual this new technique. So I think it does hold promise to kind of like as you say, show that there is something there even using this other technique that doesn't require the assumptions. And so the degree to which that can kind of answer the criticisms, the fair criticisms. I mean, these are things that we should be looking at. I think that's definitely valuable. But ideally, it would be trying to do the things that they're going to be hard to do, like collecting more data and maybe kind of changing the way we think about these things. I want to do that, but it's just going to be much harder than kind of the first. We gather a bunch of twin data, find that there's significant heritabilities for all these things that that are interesting, and look, people, this does matter. And so I think the next phase is definitely harder. That doesn't mean I don't want to do it. Okay. Oh no. Is that him? Oh. Oh. So tell him to ask James. James is down there. Ask him. So how do I? Oh my. Oh, who did that? Yeah. So I mean, twin studies and so twin studies definitely in terms of underestimate if there is gene interaction, then for sure you're not. I mean, it's going to underestimate, I guess, the so gene environment interaction in twin model. It can be done, but it's harder to actually have some measure of the environment that you care about, or have some theory about how the environment and gene interact. And there's there have been simulations that show if you are missing some gene environment interaction out there, how much is it going to bias your estimates? So you can do those, but I guess the simple linear model is definitely is not capturing all the possible scenarios. So yeah, I mean, I guess the answer is sort of yes, a qualified yes. Is that, did I answer this? Any other questions? Oh. Twin study, how we my usual way of thinking is, how much does probability of being a liberal
conservative increase in awareness, and people do genetics and don't talk in those terms. And I'd like to know whether it's possible, whether we can estimate probability of being liberal with my twin. And the question is, why don't we try to assess this genetics biology in those terms? And I would have, is there any study where you compare the impact of my parents' ideology of my genes and the comparing socialization, right? In the same study.
So there have been, you can incorporate parent data, parents and twin data. So Pete Hatimi has done a study where he has a model that includes parents. And so you can estimate, that was on one of the earlier slides, including more sibling types or additional family members allows you to estimate different things. And so you can see how much would be due to parental socialization. But the yeah, so in terms of the impact, I think that's what association studies do. Like, so they're always in well, at least in medical genetics, they tend to be represented or communicated in terms of odds ratios. You know, so this increases the risk of me getting this cancer, or you know, so it is there, but with the twin model, just variance explained, because that's this ACE model is kind of like the canonical you know, model that everyone just talks in that term, in that way for the most part. So I mean, I'm sure there could be more it could be an alternate with some incorporating some of the things that you suggested, but I don't really know of any that in terms of the twin models that do it, you know, do it that way. So it's generally just decomposing the variance into these buckets. Well, so there are a nice way to see whether or not you see the effect of a gene is you know, essentially having parents as controls in the model, and so you have like a 50-50 chance of getting your, so you know your parents' genotype, both parents' genotype, and so if they're, you know, heterozygous or they have different genotypes, and you got this type, that's kind of like a, you know, random draw. So you can speak more in causal terms, the effect of having this type of variant, and like in terms of ultimately getting the disease or like being more conservative. So there are those types of approaches. So sure, the challenge is even to collect information from families, so coming out and getting full things, getting the differences, hard to do that, not on sites. Do it.
Yeah. Keep everything solid. We're trying to go beyond it. I'm also going to